Hello. This lecture is the first of a series that will be introducing you to the various approaches that are used to study ethology. We're going to go through ultimate analysis first, and then we'll transition into talking about the various ways proximate analysis is used to examine behaviors. First, I want to review a concept map of all the different ways we approach the study of ethology. Remember that we can break most ethological studies down into one of three categories: conceptual, theoretical, or empirical. Now, the best research projects incorporate all three, but in short, here are the differences between them. The conceptual approach is best exemplified by that kin selection example I gave you. It's how seemingly disparate or disagreeing aspects of the biology or behavior of an animal can sometimes come together in new and interesting ways and lead us to break through understandings of how seemingly disconnected aspects of an animal's behavior are in fact working synergistically together. They're working together to enhance the animal's fitness. Now, there are two other approaches we can use. We can use the empirical approach or the theoretical. The empirical approach is what we just did an entire lecture on. The science lecture walks you through how to go about empirically analyzing the behaviors of animals. You gather and then statistically analyze data. You create testable predictions of behavior based on those results. And those testable predictions are your hypotheses. Remember that empirical experiments can usually fall into one of two groups, either observational or manipulative. And this is all in our science lecture. So if you want to review these concepts, go and check out lecture 2 one more time. The theoretical approach to studying behavior is a little bit different. When you use the theoretical approach, you're going to attempt to use mathematical models to either explain why animals are behaving the way they are in terms of energy budget, reproductive success, perhaps even genetic composition, or you're going to use your model to try to predict behavioral patterns and then see if they match up to what the animal is actually doing. Good example of this are what are called optimal foraging studies. These are done for predators mostly, although you can do optimal foraging studies on herbivores as well. There are several really interesting studies that have been done on pollen and nectar foraging in bees. In essence, animals tend to examine, either consciously or unconsciously, the trade-offs between what you have to do to get a food resource and what you're going to get from that food resource. When you're talking about predators, the trade-offs are between the energetic value of the prey item, what the predator is going to be eating, and then the costs involved in obtaining that prey item. There's the cost of what's called handling time. So what is it going to take to actually capture that prey animal? Do you have to chase them? Do you have to fight them? Do you have to expend a bunch of energy killing them? All of that is handling time. And another factor that's called encounter rate. How often do you actually come across prey animals? Are they rare in your environment? If they are, every time you see one, you're probably going to want to try to catch it. But if prey animals are common in your environment, you don't necessarily have to go after them every time you see them because you know another one will come along soon. Once we understand the relationships among these variables, it allows us to understand or predict the hunting decisions of a predator. So for example, the value of this fawn, this gazelle fawn, to this cheetah can be explained using this mathematical formula. Now, we're not going to go through the details, but I just want you to see an example of how mathematical formulas can be used to analyze the behavior of an animal and potentially predict it. Now, the ultimate level of behavioral analysis often combines both conceptual and theoretical approaches. This helps us to understand, first, why behaviors have evolved either in a single species or how behaviors have evolved over many different species over evolutionary time. These can be examples of what's called convergent evolution. Convergent evolution happens when a certain behavior is so helpful at keeping you alive or helping you to reproduce that it evolves independently in many different animal species that are not closely related to each other. Okay? So something like bright coloration being used to attract females. So if you're male, you're brightly colored. This attracts certain females. You have a higher success rate of mating. This shows up in birds and insects and some fish. Okay? So those are wildly different groups of animals, and yet they have all converged on this one strategy for attracting mates. It's because it's a good strategy. So whenever it evolves, it has a tendency to persist. Now, these are wonderful theories that we have. We come up with these conceptual connections between behaviors. We develop theoretical approaches to understand why those behaviors happen when they do. But the only way to really demonstrate that these connections are real is to try to come up with an empirical approach to testing the idea. See if we can generate some real data that we can analyze and determine if our theories are correct. Or if our hypotheses are correct. Now, one of the first things that we need to talk about before we really dive into how ultimate analysis is done is the difference between artificial and natural selection. Artificial selection is a tool that's often used in ultimate analysis, especially when you're looking at how heritable a certain behavior might be. Artificial selection is driven by what's called selective breeding, and this is something that we humans do. Our earliest understanding of heritability of all traits, and also the heritability of behavior in particular, came from selective breeding experiments. Now, these are really, really easy to do. In general, you take a population of a certain type of animal, and you only allow certain individuals in that population to breed. Which individuals are allowed to breed depends on what characteristics you're interested in. If you want to see how heritable white fur is, for example, in foxes, you take a population of foxes that have white fur, red and brown, and prevent the red and brown foxes from reproducing. You only let the white foxes reproduce. Then they give birth to a new generation of offspring, and maybe the majority of them are white. And once again, you only allow those white offspring to breed. Eventually, you are going to have a population of all white foxes, and pretty good evidence that the color white, or the absence of color that makes you appear white, is a heritable trait. Now, behavioral experiments have been done using artificial selection as well. The goals of these are to determine if behavior is something that's simply learned, or if there's a genetic component, and certain behaviors can be inherited from your parents. The Russian fox experiment is one of the most famous of these experiments. The Russian fox experiment was started by a man named Dmitry Belyaev, who you can see here in the upper right-hand corner, working with some of his original foxes. It's been ongoing since the 1950s. It's still happening. Currently, there are 42,000 foxes who are part of the experiment. The pups are all hand-reared. And the idea behind this experiment was to answer the question of why do so many domesticated animals have such similar physical characteristics? Belyaev noticed that domesticated animals like dogs, but also cows, pigs, even some cats, were all pro-social towards humans. Pro-social meaning they chose to affiliate with us. They chose to socialize with us. They tended to be calm, non-aggressive. All their behaviors fit the definition of what we tend to call tameness. They were all tame animals. Now, the answer to why is that we made them that way, right? All of our domesticated animals became domesticated because we started to selectively breed their wild ancestors. We were very picky about which of those ancestors we allowed to breed. So this is an artificial selection experiment that we engaged in without really realizing it. So the wild ancestors of dogs and cattle and pigs and sheep were all selected from by humans, and the individuals who were selected were the ones who were the calmest. They were the least aggressive. They were the most interested in socializing with people. And over generations, we continued to do this. We only allowed offspring who were also very pro-social towards us, who were calm, who were non-aggressive. They were the only ones allowed to breed. So we continued to select for these tame characteristics. And over hundreds or maybe thousands of generations, we started to get animals that, from birth, weren't afraid of us, were calm when they were around people and other animals, weren't aggressive. Okay? They were tame. And that's what we see today as well in our domesticated animals. But Belyaev noticed something else. He noticed that these domesticated animals, even though they were completely different species, had some physical characteristics in common in addition to their behavioral characteristics. They had floppy ears, short curly tails, juvenilized facial and body features, meaning that the domesticated animals that we were raising looked real similar to the juvenile versions of their wild ancestors or their wild counterparts. They also exhibited reduced amounts of stress hormones overall. They tended to have mottled fur, meaning multicolored fur, either in patches or mixed together. And they had long reproductive seasons, much longer than their wild relatives. This entire suite of physical characteristics was referred to as domestication syndrome. It was present in cattle, it was present in dogs, it was present in pigs, and it was present in sheep. These are wildly different animals, but they all have these two big things in common. They're all tame, and they all have domestication syndrome. What that told Belyaev and his colleagues was that by choosing for these tame characteristics, we were also getting these physical characteristics, that somehow the two were linked. So to try to figure out how consistent this linkage pattern was, Belyaev decided to domesticate an animal from scratch. He chose a truly wild animal, the Arctic fox, and started to hand rear their pups. This way, the pups would imprint on humans, recognize humans as their parental figures, and then once they were mature, the researchers would only breed the least aggressive and most tame of the individuals. And they did this over and over and over, multiple breeding seasons each year for decades. Now, if you look at the pups today, you'll see the result of all this effort. The pups are friendly towards people. They are non-aggressive. They are fairly calm. They produce lower stress hormones than their wild fox counterparts. Their ears have a tendency to flop while wild fox ears stand straight up. Their fur tends to be patchy in color, as you see on the ears, the face, down here on the paws. This is mottled fur, while their wild counterparts tend to be either bicolored, so two solid colors, or just a single color overall. These foxes have domestication syndrome. What this indicates is that these behaviors, these tameness behaviors, are likely genetically mediated, meaning there's a huge influence of genetics on these behaviors. and that these genes for floppy ears and mottled coat are somehow linked to the behavior genes, not because they help each other, but probably because they're located close to each other on the strand of DNA. So when you select for one, you just accidentally get the other as well. Okay, so this was good evidence that behaviors can be genetic. Now, how is this different from natural selection? In artificial selection, up here, I'll write it down, we choose who breeds. In natural selection, it's really the environment determines who breeds. So artificial selection is an unnatural process, but it is still evolution. You're still seeing changes in gene frequency over time. That counts. That counts as evolutionary change. The difference is what's controlling or dictating that change. The environment dictates natural selection, or the effects of natural selection, and the environment dictates which genes are going to become more common over time. In artificial selection, it's humans who choose which genes will become more common over time. Both are forms of evolution. It's just the driver that's different. Now, if you are examining how natural selection influences behavior, there's a few things that you have to keep in mind. Remember the prerequisites that are necessary if your population is going to be acted on by natural selection. Remember, there has to be variation for a trait, means you have to have multiple different versions of that trait. The trait has to be heritable, passed down from parents to offspring. And there has to be differential reproductive success. You can think of just as your fitness. Not everybody has the same success at surviving and reproducing. Now, all of these factors are necessary for natural selection to occur, but natural selection won't occur in the absence of pressure from the environment. You must have environmental pressure in order for natural selection to happen. Try to think of some examples. This should be review from lecture number 1. Think of examples of natural selection and how gene frequencies change in populations as a result of it. Now, selective pressure can act on behaviors just like it can act on physical characteristics. The example I'm going to give you is the relationship ravens have with novel objects in their environment. A novel object is an object they have never seen before. It's something brand new. Ravens are part of a group of birds called the Corvidae. Okay. This also includes birds like crows, jays, and a number of other types of birds. But what all of these birds tend to have in common is that they're very inquisitive, they're very curious. So they have a tendency to inspect new items or new things that show up in their territory. Now, there are trade-offs here. It can be very beneficial to be curious and to inspect any new thing that comes into your area or your territory. You could discover that that thing is a new food source. Okay? You could discover that it's a new nesting site that's very safe. You could discover that it's some other sort of resource that's going to increase your fitness. But it could be that that thing is dangerous. There's a risk involved here as well. It's a new object, right? You've never seen it before. It could be a new type of predator waiting to ambush you. It could be a parasite. Or it could just be dangerous. What if that bottle that this raven is looking inside of is filled with needles or broken glass. Those are things that can easily harm this bird, so it's taking a chance by inspecting this bottle. This is the trade-off that inquisitive species face. The benefit of discovery can be huge, especially if you're competing for food or resources with a lot of other strong competitors. But every time you inspect something new, there's inherent risk involved as well. Ravens take different amounts of time to approach a new object in their environment. Okay? Some ravens move at a moderate speed toward the object. They don't rush at it. They don't start flying down towards it right away. They take a little bit of time to inspect it from a distance, and then they fly at a moderate rate towards the object so they have time to examine it as they're approaching. Some ravens are very cautious. They take a long time examining the object from far away, and then they fly incredibly slowly towards the object, taking their time, really giving themselves time to fully examine the object before they get to it. But then there are ravens that move really fast towards that new object. They don't take any time at all to examine it from a distance. Instead, they speed down towards it as fast as they can. We quantify these differences in behavior as approach time. These different birds have different approach times. 30 seconds for the fast bird, 60 seconds for the moderate. That's 120 seconds under there for the slow-moving bird. So we need to ask a few questions about this behavior if we're going to put this in a natural selection context. First, is there variation for this trait? Absolutely. There's variation for approach time. Different birds approach at different speeds. All right. Now, is there an effect on fitness if you have different approach times? Absolutely. The raven that's approaching quickly is going to have high risk, but they'll arrive first, which means if there's something beneficial in that novel object, they're going to get to it first. So they'll have big benefits. So it's big risk, but potentially big payoff. Okay. The moderate moving bird will have some risk, but less than the fast moving bird. The moderate moving bird can actually sit here and watch and see what happens to the fast moving bird. If the fast moving bird gets to the novel object and it explodes, well, the moderate moving bird then dodged a bullet. But what if it's a beneficial resource and the fast moving bird gets there first? They might lose out on the benefit of this novel object. Okay. The slow-moving bird has very low risk. They are so cautious they will likely see any dangers coming long before they get to the object, but they also have much lower chances of benefiting from that object as well, because they're likely going to arrive last and all the good stuff will be gone. Let's say the novel object is a food source. Okay? The raven that's moving quickly, if they don't get killed or grievously harmed by the new object, they're going to get that food source, wind up being healthier, happier, having more babies. Or they're going to be dead if it's something dangerous, in which case their fitness drops to 0. The slow-moving bird, if we go to the opposite extreme, it's going to live a long, happy life, but it may not have access to as many benefits or as many food sources. So it may reproduce less even though it's living longer. It's hard to say. What we can conclude, though, is that there is definitely going to be an effect on fitness from this behavior. So 2 of our 3 prerequisites for natural selection to occur on this behavior, 2 of them have been met. But remember, the third is heritability. Variation for this behavioral trait has to be heritable from one generation to the next. If it's not, then natural selection can't act on it. We currently don't know, based on the information we have here, whether the trait is heritable. So how do we figure that out? We can determine how much of behavior is genetic actually using an empirical approach, right? This is actually a mix between a theoretical and empirical approach. We are going to be using a set of equations that were developed from a theoretical approach to ethology, but we're actually going to be collecting data to use with those equations, so that's how it becomes empirical. The type of experiment we are going to use to answer this question is called truncation selection. Truncation selection is a type of selective breeding experiment, so this is going to be artificial selection. And I'm going to walk you through an example of how we could use this with our current question of, is approach time a heritable behavior among ravens, right? So don't worry about these charts just yet. We're going to work through these together in a couple of slides, but just look at the theoretical approach first, as explained in the text. All right, so imagine we're working with a population of ravens that have a variety of different approach times, or what we'll refer to from now on as approach scores. Your approach score is how long it takes you to approach a novel object. So we've got a wide range of different approach scores in our population, but we are only going to allow a subset of these ravens to actually breed with each other. The way we choose which ravens are allowed to breed is we choose the ravens that have approach scores of more than 80 seconds. So these are slow movers. These are the birds who are taking their time approaching this novel object. Okay? So we're going to take that smaller group of ravens with approach scores of more than 80 seconds. We're going to breed them together, and then we're going to try to determine what the mean approach time is of those offspring. We're going to call those offspring generation 1, or X1 for short. Then we're going to compare that mean approach time of the offspring to the mean of the original population. That big group of ravens that had approach times that were both long and short and somewhere in between. We're going to call that original population generation 0. We're going to represent them as X0. Okay. The first factor we're going to calculate is something called the selection differential. We're going to subtract the mean of our original population from the mean of our offspring. And that number is going to represent the total amount of variation that could be explained by evolution alone, or by natural selection alone. Okay? So if you are only relying on natural selection to affect your approach time, you would see the selection differential would tell you the maximum amount of a change that natural selection could actually do by itself. An easier way to think about this is if an animal's approach time is 100% dictated by genetics, that difference between X1 and X0 is the amount of change you would expect to see. If genetics is controlling approach time, then the genes present in our smaller group of slow approaching ravens would be dramatically different than the genes present in our larger group. And those genes would be the only thing influencing approach time. And the selection differential would be an actual measure of that. But we're not done here. The next thing we're going to do is we're going to take the offspring of those slow approaching ravens, our first generation, and we're going to breed them together. The offspring from generation 1 will give us generation 2, which we'll call X2. If we then subtract our original population's mean from X2, we'll get what's called our response selection value. The response selection value actually tells you how much the truncation experiment itself has affected behavior across your generations 1 and 2. Okay. If you get a high response selection variable, it means that limiting the genes that were present in our artificially selected populations, meaning limiting the genes in our group to only those held by ravens that approach slowly, it had a big effect. So a big response selection number means that limiting the genes in our gene pool had a huge effect on the resulting behaviors you see in their offspring. If the response selection variable is low, it means that limiting genes did not totally dictate the behaviors of the resulting offspring. Okay, so high value means that limiting your genes, let's say limiting your gene pool, had a big effect on your behavior. But if this number is low, that means limiting your gene pool to just those slow-moving ravens had little effect on the behaviors of their offspring. We can actually use our selection differential and our response selection variables to calculate or estimate how heritable the behavior of approach time actually is. This all makes more sense once you start to work through an example. So let's do that now. Let's put this in context and actually put it to work. All right, so we're going to start with our original population of ravens. Okay. So let's draw a couple of charts here. Okay. This is going to be our original population. Number of ravens over here on the y-axis. And then on the x-axis, it's going to be approach time or approach score. In our original population, we have pretty much a normal distribution of approach scores. So some ravens approach very quickly, so it doesn't take them much time to approach the new object. Some approach slowly. It takes them a long time to get around to getting to that object. Okay. Now, our average approach score for our original population winds up being 60 seconds. Okay. So our x0, which remember is the mean for the original population, that's 60 seconds. Okay. But we are going to limit what birds in this population are allowed to breed. Only birds with approach times of greater than 80 seconds are going to be allowed to breed, which means only the birds in this part of the curve will be allowed to reproduce. Nobody on the left side of that line is going to be allowed to reproduce. So the only genes being passed down will be from these individuals who have really slow approach times. In fact, the mean approach time for this smaller group of ravens is right here. It's 90 seconds. That's the mean for our fast moving, or for our subset of the original population that's actually going to be allowed to reproduce. Now, what you see next is going to be the offspring created by this group. Okay. So if we only allow individuals with these slow approach scores to reproduce, here's what their offspring are going to look like. So same exact graph showing the same data. But what we see now is a much more skewed curve. The mean of this curve is actually 90 seconds. Okay? So this is no longer our original population. This is now generation number 1. These are the offspring of the birds that had approach times greater than 80 seconds. The average approach time of these offspring is 90 seconds. So that's definitely different from our original population. So from this difference, we can calculate our selection differential. The selection differential is calculated as S equals x1 minus x0, or x0. You always subtract the original population from the newer population. Okay? So what does this give us? Well, x1 is 90 seconds, x0 is 60 seconds, which means that S equals 30 seconds. That's the maximum amount of change that you can account for through natural selection alone. So if this behavior is 100% genetic, you'd expect to see a 30-second difference in each new generation. Now that the population's distribution looks like this, all the genes that you have in your population are represented on this graph, you would expect generation number 2 to look just like generation 1. Another narrow curve biased towards the slower end of the scale. If the parents are slow approachers and approach time is 100% genetic, they're going to pass on those slow approach genes to their offspring, and all you'll have from now on is 90-second or more slow approachers. That's if this behavior is 100% genetically controlled. Now, how do we verify if it is or if it isn't? How can we test to see if the behavior is truly just genetic? Well, we're going to do one more round of artificial selection. This time, we are going to allow generation 1 to breed with itself. No constraints, no restrictions. Any bird in generation 1 can breed with any other bird in generation 1. And that's going to give us our generation 2. So the offspring of generation 1 have an approach time distribution that looks like this. The mean approach time for generation 2 is 70 seconds. So remember, mean approach time for Gen 1 was 60. For the original generation was 60. For generation number 1, it was 90. And for generation number 2 now, it's 70. So that's x sub 2 equals 70 seconds. Now we need to calculate our response selection. Okay. Response selection is calculated as R equals x2 minus x0. So not x1, but x0. We're still comparing the effects of our truncation of the population to the original natural population. So remember, the original population is what these birds are supposed to look like when it comes to the distribution of approach times in this group. What we're trying to see is if by truncating the population to just those who are slow approachers, can we change the approach time distribution permanently as it compares to the original population? So you're always going to be subtracting the original population from the younger one. So our second generation approach time is 70 seconds. Our original population approach time is 60, which means that R equals 10 seconds. Remember, this represents how much truncation has affected behavior across your new generations, meaning how much limiting your gene pool affected the traits you see in your population. This is not a very high number. 10 isn't that great, or not that large. But here's the real important calculation, the one that you're actually going to use to draw conclusions from. Heritability. Oops. This calculation is going to actually tell you how much of this behavior is truly heritable. Heritability is your response selection variable over your selection differential. So in this case, that's 10 over 30, which is 1 over 3. 1/3. 1/3 is how much of this behavior is actually controlled by genes. So only 1/3 of the variation that you see in approach times is actually genetically controlled. The rest is controlled by something else. So only 1/3 of it is heritable because only 1/3 of it is genetic. So if a behavior isn't all caused by genes, then what is it caused by? What else is influencing this behavior? It can be either environmental factors and or learning. You might learn a different approach strategy from a member of your flock or from your parents, one that's slightly different than the one that you are inherently born knowing how to do. There might be effects of diet or upbringing. You might find early in your life a new food source, which changes your search image for foods from that point on. So if you encounter a new novel object, but it vaguely looks like this strange food you encountered as a chick, you might be more inclined to approach it rapidly because even though it really is novel, it has enough in common with something you've seen before that it seems safe. So your approach time might be faster. So different environmental factors and learning factors also affect behaviors. But some of every behavior is also heritable. Or not every behavior, some of most behaviors is also heritable. Usually it's only about 0.1 to 0.4, so about 10 to 40% of the behavior is genetically controlled. The rest is influenced by experience, environment, learning, what the organism goes through during their lifetime. Now, if at least some of every behavior is genetic, you would expect there to be a strong correlation between parental behavior and offspring behavior, because offspring are inheriting their genes from their parents. Now, to test this, you can do what's called a parent-offspring regression analysis. Okay? You directly examine in these experiments the correlation or the connection between parental and offspring behavior. If there's high heritability for a certain behavior, you'll get a high correlation, regardless of what other factors are present, regardless of environment, regardless of learning. If genetics are the primary dictator for a behavior, you're going to see a huge overlap between parental behavior and offspring behavior, even if the two live in totally different environments. A classic example of one of these studies was done on cliff swallows and their group size. So cliff swallows are social. They build these beautiful mud nests on either the sides of buildings, if we happen to construct a city near their natural nesting grounds, or they'll build these mud nests on the sides of cliffs overlooking bodies of water. And the nests are built from the edges upwards. They lay layer after layer of mud, getting narrower and narrower until they create an opening that's just big enough for one parent to look out of. These mud nests are really strong. They'll fill them with soft straw or grass, lay their eggs in them, raise their offspring, and then fly away until the next breeding season, where they come back and rebuild the nests which have fallen apart. They do live in social groups, and as you can see, the nests are pretty close together. You can have rows upon rows of nests built above and below a certain bird on a cliff. So these birds might have multiple rows of neighbors above them and multiple rows of neighbors below them. That's a lot of cliff swallows all in one place. The size of a group of nesting cliff swallows has effects on their individual fitness. This actually creates selective pressure based on their group size. Crowded groups, or just big groups, big groups experience a problem with parasites. Parasites spread easily among densely populated groups of cliff swallows, and they can have major effects on swallow health, and they can even kill chicks as they're developing. But small groups, while they don't have the problem with parasites, do have a problem with predators. Predators are more likely to attack small groups because they have fewer defenses, and small groups can be wiped out by predators. So there are pluses and minuses to living in big groups or small groups. There are advantages and disadvantages to both. So the behavior question here is, if offspring live in groups that are sized similar to their parents' group size, is that choice genetic or environmental? So if offspring are choosing to live in large groups and their parents were also large group nesters, is that a genetic connection, or is it something about the environment that's leading them to make that choice? To examine this, we're going to do something called a cross-fostering experiment. This is where you take offspring and you switch them between different parents. So we'll start with a set of large group parents, and we're going to give them offspring that are originally from a small group parent. Okay? We'll then take large group offspring and give them to small group parents. Okay? That's the vice versa part. So you've got large group parents raising small group offspring and small group parents raising large group offspring. The idea here is that if group size choice is 100% genetic, then it won't matter if large group offspring grew up in a small group. When they're mature and they get to make their own choice, their genes are going to tell them you need to live in a large group, regardless of the fact they were raised in a small group. Okay? But if environment, learning, and upbringing are more important than genes, then those small group offspring that were raised by large group parents are going to switch and become large group nesters as well, because that's what they were raised in. That's a situation of little to no genetic influence. What you see when you do this cross-fostering experiment is that the pattern of preference in offspring is unchanged. What that means is that even though small group offspring were raised by large group parents, they still nested in small groups when they were adults. Large group offspring, raised by small group parents, likewise ignored their upbringing in the small group and chose to live in large groups as adults. This indicates that there is a strong genetic component to determining this behavior. Genetics in this case trump upbringing and they trump learning. There's one more topic I want to cover. It's the connection between evolution and behaviors associated with something called sociobiology. Sociobiology is the study of the evolution of specifically social behavior. Animals that live socially may be actively cooperative, sharing food, sharing responsibilities, but that's not a requirement. Just living in a group, living near others of your own species is enough to be considered social. Guppies are a great example of this. Poecilia reticulata is their scientific name. They're a model organism, meaning that they have been studied extensively by scientists. We know a whole lot about their biology, which means that we can ask complex questions about them and already have the background to help answer those questions because we already know so much about them. Wild populations of these guppies are found in streams, these stream habitats in Trinidad. And the streams are separated by rocky waterfalls, so you get separate populations that are isolated from each other. This means there are different environmental and different social or community conditions in each part of these streams. This is going to affect guppy behavior differently. Now, guppies are small and tasty, so they have a fair number of predators, which also means they've got some pretty good anti-predator strategies, ways to avoid being eaten. Now, guppies live either in upstream or downstream populations. Up, down. Upstream populations are only exposed to really small predatory fish. Those are the only predators they have to contend with. It means they're under low predatory pressure. They're not dying in large numbers because of these small predatory fish. Some guppies are lost to these predators, but not huge numbers. Now, downstream populations are a different story. In downstream areas, there are numerous large fish predators. This results in a high predator pressure in those regions. Lots and lots of guppies are getting eaten by these bigger and more numerous predators. Now keep in mind, there's almost no gene flow between populations. So the waterfall that separates these two groups really does significantly separate them. Different traits are going to be selected for in these two different populations, and those traits are not going to be shared because the two populations don't interbreed. So let's take a look at what this looks like. Okay. So here's our upstream group and our downstream group separated by our waterfall in between. Now remember, in our upstream environment, we've got little predatory fish. Okay, so that's our predator. We've got small predators that are not super common. They're around, but there aren't overwhelming numbers of them. And then in our downstream environment, we've got a different situation. Downstream, we have lots of big predators. So lots of big, hungry, predatory fish. All right. Actually, move this over here. So this is our predator situation. So how do our guppies deal with these predators? Guppies have three primary anti-predator strategies: shoaling, breeding, and growth. Shoaling is when large numbers of guppies all hang out together. They form what's called a shoal. They do this because there's safety in numbers, and you more commonly see this in high predation sites, which means shoaling is going to be really common down here in our downstream population. Next up, breeding strategies. High predation sites breed faster, they produce smaller offspring, and they energetically prioritize reproduction. It means that they reproduce when they're younger. And they have many offspring because they're putting all the energy they can into making them. All right, so at high predation sites, you have breeding strategies that include early reproduction, and lots of small offspring. Now, at low predation sites, you see a different strategy. Instead of rapid breeding, what you see is emphasis on growth. At low predation sites, guppies have fewer, larger offspring, and they develop rapidly. It means they grow fast. Okay. So in our upstream populations, with low predator pressure, you have fewer, larger offspring. Actually, they're just large offspring. Okay. And they grow up fast, grow to maturity real fast. Okay. So what does this mean? Why is this important? We'll just look at the patterns, and we can explain why these behaviors exist. When you have small predators, one great way to survive them is to be the same size or larger. So I'm going to draw our guppies here in green. Okay. So if you start out as a teeny tiny little guppy, but you grow very, very fast into a nice large size guppy, you're going to be too big for this predator to eat. Okay? So if you're already born fairly large, you've accomplished the first couple of steps of your growth already. So if you're born already about this size, it's not going to take very many more growth stages for you to get to a size where you're big enough to avoid being eaten by the predators that are around. It makes sense for offspring to be born larger and then to grow up fast. Size is your best defense against a small predator. Guppies upstream have fewer offspring because they have to invest more energy in each individual offspring because they're so big when they're born. So they can't have as many. Right? Now, if you're downstream, you got lots of problems. Lots of predators. All of them are pretty big. So here you're going to have to employ a lot of different options. You're going to have to engage in shoaling behavior. So just hanging out in large groups so that hopefully if somebody has to get eaten by one of these approaching predators, it isn't you. Okay. And you're going to reproduce young. Why reproduce young? Because you want to reproduce before you die. And the odds are your life expectancy is going to be pretty short in a high predator environment. If you want to pass your genes on, you need to reproduce fast, early in life. Also, you're going to produce lots and lots of little offspring. Tons and tons of them. The reason for this is the odds of any single one of these offspring surviving is pretty low. With this many predators around, a lot of your offspring are going to get gobbled up before they have a chance to grow up. You produce tons of them in the hope that at least a few survive. Because you're producing so many, you can't invest that much energy in any individual one, which is why they have to be small. Okay. Now, you can do a lot of interesting things with guppies, but in this context, we're going to be looking at their social behaviors, right? Especially shoaling and these mating behaviors, when they mate and the kinds of offspring they reproduce. Now, to test the effects of environment on social behavior, you can do something called a reciprocal transplant experiment. This is where you move subsets or small parts of your populations to different sites and measure the effect that new environment has on subsequent generations. So in this case, we're going to take upstream guppies and we're going to move them into a downstream habitat. That's called a transplant. When you pick them up and move them to a new place, you're transplanting them. But remember the reciprocal part of the name? Reciprocal means fair. So you're also going to take some downstream guppies and move them upstream. Okay, it's going to be balanced. Researchers took guppies from the high predator sites, okay, and put them in the low predator sites, and vice versa. And what they found was that when they reversed the selection pressures like this, suddenly low pressure guppies were experiencing high predator pressure and vice versa, they saw some really interesting effects. Over time, behavioral reversals were observed. What does that mean? It means that guppies from low predator sites that were transferred to a high predator site took on the characteristics of the guppies that had always lived in those high predator sites. Guppies from the high predator site, when transferred to the low predator site, also over generational time took on the characteristics of guppies that had always lived in the low predator site. They changed. You saw behavioral changes to match the environmental pressures, which makes sense, right? If they couldn't adapt to these new environmental stressors, they would just die out. But this is evidence that social behaviors are subject to evolution, and they're subject to pressure from the environment shaping their evolutionary trajectory. All right, that's it. I'll see you next time for proximate analysis.